My name is Will Wright. I'm the Chief Operating Officer of Chonex. We are a soil health company working to upcycle natural agricultural byproducts into high value solutions to improve soil health. Um, we've been at this for several years and starting to see some really great results. Pleased to be joined here with by Misha Vandal. Misha has been someone we've worked with over several years. Misha runs Vandal Land Management, where he assists vineyard managers and wineries in developing sustainable regenerative agricultural practices, reducing chemical inputs, and increasing biodiversity. And really, Misha was an early advocate, an early sort of person who understood what we were working on and understands the value of a lot of this. And we've asked Misha here because of his experience with our product, but also just generally his experience helping vineyard owners and managers transition to regenerative and more sustainable practices, really to share his points of view on why that's an important why soil health is so important in vineyard management and growing specialty crops, what some of the struggles are that he's seeing and some of the solutions that he's bringing forward. So this will be educational as well as a discussion of some of the reasons why Misha is working with us, but really intentionally here asking Misha to share his points of view with everyone here. So again, if you have questions, you can place them in the chat. It's not a huge audience here. We've got about 100 folks registered, but so they'll come and go. But we will be sharing this also afterwards for posterity. So Misha, really, if you want to take it from here, that would be great. And let's go. So,
All right, sweet. Thanks a lot, Will. Like Will said, my name is Misha Vandal. I studied microbiology at Old State University, and early on, I worked in several of the labs at the college there. We did one of the labs was a medical microbial lab studying the uh, intracellular symbionts between a tick host and a rickettsia. So I did a lot of DNA extraction and stuff in the lab, and working with PCR and stuff. And that pretty much drove me out of the lab and moved me towards working more in agriculture. One of the other labs I worked in was with extremophilic bacteria, studying origins of life and horizontal gene transfer, what triggers bacteria to pass their genes horizontally through environments. And that was really interesting. And then I kind of took some of that knowledge from microbial ecology and applied it to agriculture and started working with compost teas and regenerative practices, permaculture, and helping farmers kind of adjust their nutrient management programs to incorporate some of those practices. And one of the biggest challenges with that is finding a practical solution to apply compost tea, which is kind of a labor intensive process to brew a tea and then get it out. And so we've been looking for solutions to accomplish those goals and get a large diverse microbial community applied to a soil and enter Chonex with strong soil, and they have a really unique product. So you're excited to start working with it. On the vineyard management side, I've been working with vineyards for about 5 years now. I live in Napa Valley, and the last year we did some pretty broad trials across 13 different sites with strong soil, doing different application windows and different targets for success. And so we're going to move right into it. Strong soil on wine grapes. What makes it unique is that strong soil provides kind of a entire microbial community as opposed to a single inoculant. So it's you're more likely to get successful colonization and inoculation with a broad spectrum of a community as opposed to an individual inoculum. You might see random success with a single inoculum, whereas you're going to see kind of a microbe move into an existing community more successfully. So going into it, why wine grapes? Wine grapes are a unique crop, and they are a very technical specialty crop. We gather a lot of data. We have a lot of um, focus on soils, and because of the fact that we're not farming anything from seed or anything, it's a lot different than any annual crop because there's so many implementations that you need to take into account with choosing a rootstock, choosing your clone, and then across the different varieties, what makes the perfect match for a rootstock and a clone. And the soil, it's there's a lot of thought that goes into it, and a lot of data that's collected, and sometimes you don't always hit the mark. So um, we're still learning a lot as we move forward. There are growing problems in the wine industry, in viticulture and vineyard management. Um, so that's kind of why I thought it's a great model organism for these types of research, is that we have a lot of data dating back multiple years on a lot of these vineyard sites, and we're seeing different variations of signs of soil distress, right? And things where plants that have been thriving for 6, 7 years, maybe as many as 25, and there's symptoms of decline going on. So I think everybody in industry is starting to open up to the idea that the one area that hasn't really been exploited is the microbial side of things. And a lot of that's due to we really haven't had the ability to do mass metagenomic testing on soil samples until probably 4 or 5 years ago, where it became more economically feasible to do so. So now that things like biomakers, where we send our samples to, are available to farmers, we can really start to dig into the details of the microbial communities that are representing each soil site. So moving into more of that, we're going to kind of go over current grower concerns, what we saw during pre-treatment testing, what our desired outcomes were, and then our post-treatment results, and what kind of conclusions could we draw from that. Some of the major grower concerns are going to be viral diseases, heat, drought, water stress, climate-related issues. We saw last year it was super hot here, for instance, and you could see it on the plants, and the canopy was struggling at certain points, especially once it gets over 100 degrees. It's really hard to have successful biological reactions within the plants or the vines. So, how do we mitigate some of these stresses without adding water? Water is a big concern for vineyard management. We don't want to over irrigate because that leads to a whole myriad of other problems. So, and then beyond that, there's insect pests that are triggered by plant health decline symptoms, and things like heat spikes that stress out the plants, create vulnerabilities for insect pests to exploit. So then moving beyond that, there's the fungal disease side of things like trunk vine canker or grapevine trunk diseases.
Hey, how you doing? Uh-huh. You had a good lunch?
Oops, oops.
And then beyond the fungal diseases, there's the soil health. So viral diseases currently impacting, I'm sure most people who are in the wine industry or in the viticulture side of things on this presentation are aware that grapevines are susceptible to several known viruses, causing major economic losses. The top one right now in Napa Valley, for instance, is grapevine red blotch. What the problem with that is that the vines will be growing just fine, and then when we get to the end of the season, they kind of stall out, and the ripening is pretty much paused, and we can't really utilize, if the symptoms are so bad that the Brix doesn't achieve the desired metric, then we can't really even use the fruit for winemaking. So we're starting to see more and more vineyards get torn out, and we're replanting. And some of the ones that I'm working with right now were replanted 5, 6 years ago. They're pulling their first crop right now, and there are already symptoms of grapevine red blotch moving right back in. So how do we identify, like, beyond just the vector, that we know that there's an insect vector, we know that um, you know, there's plant signaling hormones that draw those insect vectors in, and what moves this virus through vineyards so aggressively. It's currently widespread in the Napa Valley and through the Central Valley, and it's a huge economic concern. And one of the big areas that people are starting to indicate as a causation is soil health, right? So if the soil health isn't intact, then you really can't get beyond that and get a successful crop in. And the more distressed the soils become, the harder it is for a replanting to be successful. Heat, drought, water stress, and climate. The emerging challenges there are the weather gets more and more unpredictable, and things get more and more exhausted as we're doing our aggressive farming practices, such as tilling and chemical fertilizing, pesticide use. All these things stress the plant out and the vines out in general, and then you couple that
Hello?
with heat stress
Yes.
and climate stress, we get a lot of kind of a cascade, or a grouping of impacts that just start to deteriorate rapidly. So the insect pest that we're really concerned over right now is the three-cornered alfalfa hopper. It's the vector of red blotch, and chemical control for this particular insect is not recommended. So you really, they're not even really present till later in the season, and usually we stop spraying mid-season. So I really feel that some of our best defenses against this pest is to create a soil environment and that's very supportive of plant health and resilience. Insects only go after plants that are under distress and signaling that they're ready for a state of decay. Insects tend to be, they are, cyclers of biology. So if your plants are healthy, theoretically, you shouldn't see any insect pests. So how do we build the soil to avoid these leaf hoppers to come in and wreak the havoc that they're currently doing? So that's kind of where the importance of soil microbial communities comes in, is that if we're aggressively farming the soil and we're tilling, creating soil compaction, where everything we do in industrial agriculture kind of is not counterintuitive, but somewhat negatively impacts microbial communities. It's one thing that's never really been able to be studied, but everything that's coming out with the current research is that a lot of these practices negatively impact these communities, and you can see downward trajectories over years of these programs. So that's why the biofertilizer industry is kind of starting to take off, because it's pretty overwhelmingly obvious that we can't neglect such a large component of agriculture. Introducing a product like Strong Soil, which is rich in the beneficial microbes, can effectively jumpstart the soil's natural biological processes. These microbes help break down organic material into forms that the vines can readily absorb. It can improve nutrient availability and soil structure. Over time, as we revitalize the microbial community, it'll improve soil health and the resilience of the plants. This means the vines will basically become more robust, better tolerant of climatic conditions and weather events and so on and so forth. Fungal diseases that are persisting in the vineyards are grapevine trunk diseases. They're composed of roughly 32 genera of fungi. One source stating there are over 133 species across 34 genera that target and exploit vulnerabilities of vines during states of either heavy rainfall or drought. So we have drought season, then we have a heavy rainfall season, and these swings in climate can create stress on the plants that are then exploited by these fungal diseases. And we're starting to see more and more fungal diseases that weren't necessarily considered too much of a problem 5-10 years ago. Things like Bacillus and Phomopsis are kind of becoming a little more prevalent, where UTEPA was more of a concern 5-10 years ago. And then the soil health aspect, there's so much to it that it's really hard to really plan your whole season around soil health. So I feel like preventative maintenance and preparing your soil microbial communities for whatever the environment might throw at it is going to be better than trying to course correct post a catastrophic event like a major heat wave or a prolonged drought, things like that. You're better off incorporating some of these practices as part of your baseline to support the plant health so that they're well prepared for these types of events. So moving forward. Poor soil health is costly. We're ripping out probably about 10% of the vines in Napa Valley this year, and there's talk of another 10% next year, which is very, very costly for any vineyard or landowner. It costs a lot to replant. The labor is intensive, and if you don't correct the cause of the issue that drove you to have to replant your vineyard, you're just going to end up with the same symptoms shortly after. As I explained, we replanted one vineyard 5-6 years ago. We're just pulling the first crop off it, and we're seeing 10% of the vines already showing symptoms of red blotch. So give it another 4 years and it's going to have to be pulled out again. And that's basically, there's no economic gain on that. Before you even see a return on investment, you're having to spend the money again to replant. So moving into the strong soil, we did some pre-treatment testing all around the valley. And I'm not going to get into every single site that we tested. I'm just going to focus on a couple of the sites that we really have results on. We're still waiting on actually results from the biomakers on about 6 or 7 of the vineyards. So, but pre-treatment testing, we gathered the data and we tested an organic site and a conventional site about last July is when we kind of started talking about really trying to tackle this issue. Both sites were low in carbon. They had low biosustainability. Soil health was in need of improvement. And when you looked at like the structure of the communities that were there were an abundance of pathogens present. They weren't necessarily impacting the vines, or the symptoms weren't necessarily evident, but there was definitely a large presence of fungal trunk vine diseases, Bacillus, Phomopsis, Blackfoot disease. All these organisms were red flagged as being in concentrations that aren't really favorable to a healthy vineyard, or they're indicative of a soil that's vulnerable to these types of diseases. And that goes back to like tilling. If you're going to be aggressively tilling, you're going to be decimating the beneficial fungi populations, which basically leaves the door open for either non-beneficial or even harmful fungi populations. Um, so the desired outcomes that we were really looking for were trying to skew these microbial communities into um, more of a beneficial standpoint, really. So we wanted to see some depletion of some of these, negative players and some of the key players, and we wanted to increase the populations of beneficials and keystone species that promote um, plant function, like phosphorus solubilizers and um, plant hormone producers, phytohormone producers, and drought tolerance hormones, and soil structure, polysaccharides, and stuff like that all contribute to the benefit of the soil and can help cycle nutrients and increase the abilities of the plants to be successful. So we dove in and started this year. We did about 5 rounds of treatments, um, all mostly with strong soil. We didn't really tweak too much on the existing regimen or feeding program that they've been running for X amount of years. We didn't want to make too many changes because you can't really pinpoint what you're doing if you make too many alterations to a current program. So we focused on just a few minor adjustments, mostly just adding in strong soil, maybe drawing back a little bit on some of the chemical fertilizers and even the organic fertilizers, just kind of scale it back about 8-10% to accommodate the addition of strong soil. We know we don't want to overfeed grapevines. We don't want to have too much vigorous canopy. We don't want to have heavy crop loads. We in the uniqueness of grapevines is that when you're trying a product like this, we're not really like the metric that most farms are going for is going to be yield increase, right? And in viticulture for wine grapes, we're really targeting our target yields are set to 3.5-4 tons per acre. Anything beyond that is going to negatively impact the quality of the wine. So we can't really tell the success of a product based on that, but we can see increased numbers of clusters and how the size of the clusters, berry size, all these metrics that we can kind of track while we're doing it, while we're treating these vines to see their success rate. So moving forward, some of the desired outcomes were going to be the metrics we were looking for were yeast assimilable nitrogen, hormone production, carbon increase, and phosphorus solubilization increase. So the site that I'm talking about had about 10 years of data, 0% or 0% yeast assimilable nitrogen on this crop. The vines are about 25 years old, and as long as the winery that I'm working with has been sourcing grapes from this site, they haven't been able to achieve any yeast assimilable nitrogen. And for those that don't know, that's a metric of nitrogen that's in the fruit that yeast uses to get nutrients and proliferate to make the wine. So without yeast assimilable nitrogen in the grapes, you have to add it in the winery, and it's kind of a metric of whether or not the grapes are that quality. The less stuff you have to add in the winery, the better, the more representative the fruit is of the terroir that you're pulling it from, or the soil site. So We wanted to increase the use of soluble nitrogen in this particular site. That was one of our target goals there. So moving into it, we saw, so this is some of the data on the microbial shift that we saw. Post-treatment, you can see these are the box clusters from the biomakers, the tools that they have on their website that when you send your samples in, you can run against each other to see. So cluster 2 in this group is the pre-treatment, and cluster 1 is post-treatment. So rhizobium species, we saw an increase of about 10%, 15% increase in that. And Pseudomonas putida, we saw about a 44% increase. Pseudomonas putida is a bacterium known to solubilize phosphorus, can boost iron availability, and improve soil structure. The reason I cited these two is that the research behind the benefits of Pseudomonas putida are well established. It is frequently a product all on its own. There are plenty of bio-inoculants. I don't know if they're considered biofertilizers, but they're biological inoculants of just Pseudomonas putida. You see them usually with about 2% Pseudomonas putida, and maybe it's either in water, inner ingredients is water or dextrose or some sort of carrier agent to get it out into your fields. Strong Soil has Pseudomonas putida present in its community, and I feel like being able to apply it with other as a community inoculant, or instead of trying to apply it singularly, you're going to have a better success rate for it to establish itself. And a 44% increase was pretty significant considering the state of the vineyard beforehand. The next that we had was bacillus species. There are a few bacillus species in the Strong Soil product. I think there's 3, and overall we had increase of about 158% in detection of that particular species group. And again, bacillus is a well-researched, well-established beneficial microbe in the soil, and to have that increase is a pretty good indicator that we had a successful inoculation with the product. And then on the other side, cladosporium, which was flagged in our pre-treatment testing as a potential harmful soil fungi. It's kind of ubiquitous, cladosporium. It's found all over most soils, but it's really never considered to be a problematic individual unless it's in very abundant populations. So in this particular test, it was relatively abundant on our pre-treatment testing, and then post-treatment, we were able to reduce its population by about 58%, which was pretty significant indicator that the Strong Soil and the treatments we were doing were having an impact on the microbial community, and not just like a positive impact. So, and then on this site, the YAN increase was pretty significant. The bar graph shows that the last 5 years, it's been 0 across the board. And, this year, for the first time in the winemakers' knowledge of the grapes, they were not only over 0, they were at 80. And my target for that was about between 60 and 100 YAN, which is a measurement of milligrams per liter. And I think if we do a replicate of this again next year. We might increase the dosage on strong soil and couple in some other biostimulants to kind of help catapult it into maybe 150 target yan, which is really where the winemakers want to see their yan at. 150 to 200 is pretty standard. If you go over that, it can cause other problems in the winemaking. So moving forward, these are some of the soil community screenshots. Okay, if you look at the one on all the way on the left over here, this is the pre-treatment test, and you can see that the big large bubble there is actually a Cladosporium sclerotia. And when you're talking soil microbial communities, you want to see something more like the center community there, where you have lots of more biodiversity, but no one individual is like dominating the population or dominating the community. Anything where there's a huge dominance of one particular organism, it's an indicator that something's not right or that there's a bloom in that particular organism. And if it's not necessarily a beneficial bloom or an expected bloom, like you might see during spring when the plants start waking up, it's not necessarily something you want to have happen. So the first pre-treatment tests showed an abundance of Cladosporium and some harmful bacteria and harmful fungi. The other ones were Alternaria, which is known to cause problems, and then Aspergillus was a big one that was detected as well. And as we did our treatments, the test sample we pulled recently, all of those individual populations within the community were downsized so that we got a better balance of individual populations comprising the entire community. So what type of conclusions can we come from this? Can we derive from this? Clearly, there was a positive impact when we applied strong soil and when we applied these techniques to the farm. The yan was increased, which was a pretty solid indicator that we had success in what we were doing. And the other thing is that there was no real negative impact, right? So a lot of people have concerns about there's a lot of farmer skepticism on using these products or changing their current feeding regimen to incorporate some of this stuff because, they're outside of it being successful, there's the other side of the coin that it might cause a negative impact. And, nobody wants to explore something that they're unfamiliar with, or there might be a risk, a negative impact. So I think through doing these trials, the trust has been expanded upon to potentially not only try it on more fields, but increase the usage and potentially do some more foliar applications and stuff like that. So really, I think it was a pretty, great success that we didn't really have anything negative happen. And given the circumstances of the vintage or the year, it's been kind of a struggle. The weather hasn't been great. It's been hot and then it's been cold and then it's kind of been all over the place. So we're seeing a lot of variations between different sites, and the variations are less dramatic between the strong soils treated sites, which kind of helps create some predictability. So yeah, that's pretty much where we were at and how far we got with the whole program. Things that I would do differently maybe next year would be timing and trying to get it out on time. Some of our timing might not have been as ideal as I would have liked. There's a lot of moving parts in a farm, and when you're not the sole person in control, it's hard to remind the owner of the vineyard to remind his vineyard manager to remind their laborers to apply a product. So it can get, lost in translation, and sometimes you got to make sure that the product that was dropped off, say at the farm, is actually getting out into the field. So I think having the successes that we did, it's going to be a little bit easier to kind of jumpstart the owner of the vineyard to really put the pressure on his team to get these products out there, and we'll hopefully see increased results. And we're going to do some more testing time points. The only drawback to that is that none of these farmers are really looking to add more costs to their production. They just really want to know the product works. They don't want to have to test for it. They just want to make sure that it rolls out without a hitch, and it's a slam dunk. So I'm definitely more confident that we can do that next year. So we're trying to push to really get some more strong soil on some more vineyards. And that's about it.
Awesome, Misha. If you've got some time, we've got maybe some questions if folks want to come off of mute and ask. Thank you. And so, Misha, that was great. I know that you're a believer in the products. I really appreciate you talking about this. I think I had one question, and this is more for, obviously we want to sort of be able to deliver a high quality solution to farmers. But you had talked a little bit about some of the other ways that you've tried to restore biology to soils, whether that's compost teas or other products.
Yeah.
Can you talk a little bit about why those work, but also why they might not work, and some of the advice you might have for farmers considering those?
Definitely. Compost teas are great. They have plenty of back data to support their use. I think that the biggest setback for them is consistency. You might not always get the same microbial community unless, even if you do the same thing the same way every time. Things like time of day or time of year, or the inputs that you're using, what types of manure or types of fish emulsions, whatever it is you're using. There's so many variations that can go into play that it's really hard to pinpoint that which product or which additive to a tea is the catalyst or the major benefactor, the major beneficial component. So on a large scale, in your backyard garden or something small, like 10 acres, I mean, yeah, it's doable and you can get consistent results. But when you try to scale that up to 200, 500, 1,000 acres, 25,000 acres, it's borderline impossible. I mean, you can't brew a tea for 1,000 acres without it getting inconsistency within the tank. Like the tank's got to be a certain size. You have to have pumps and all these things to get it to work. And then you might not know exactly what you did. And just the labor component alone, I don't see too many farmers, unless they have the infrastructure set up to do that kind of program, to really find the time to do it. I mean, a lot of farmers are already overwhelmed as it is, and it's always a race against the clock to get everything done before the sun goes down and the season's over. So I just feel like that's been the main deterrent for farmers adopting these types of programs into their feeding programs. So having a product like Strong Soil, where you can just toss the biology and agitate it and it's ready to apply in 15 minutes, is a huge game changer. And really, the farmers that we were working with that used it were really receptive to it. They didn't have to supervise their staff to get it out. And it's a consistent product. Other products that are comparative, the reasons why I'm not 100% sold on some of them is that they're not exactly whether they're shelf stable. You can have products that are, like I said, single source inoculum, single species bacteria, single species fungi, and you can do everything you can to get it out. But when you're adding, say it's a mycorrhizal fungi and it's suspended in dextrose, it's 2% biology and 98% sugar essentially, and you're trying to um, agitate the tank and get it all out there. There's definitely some inconsistencies there, and there's definitely been some research that there's other biology in there because they usually add those sugars and stuff post process. So they get the biology content based on their concentration and how they dilute it down, but whether or not their sanitation practices, there's risk of contamination in there. And so the odds of successful inoculation, I feel, go down with those products. And that's why you get a lot more variations and success
Yep,
stories on those. So,
that's helpful. And actually, and Tom sort of asked a related question. I mean, he's asking, is there an advantage to add fish emulsion and molasses when applying a microbial microbial mix? What are your thoughts on kind of adding, something like a molasses or a fish emulsion while
yeah, I would say, those it's great. Those are definitely beneficial. If you think of strong soil or the product as like your base biology, and then you use, it's just like you give the biology a little bit of food, or you're stimulating that biology. So some of the treatments we did strong soil application and then followed it with a strong soil application with a little bit of liquid biochar and a little bit of soy hydrolysate to kind of stimulate the biology that strong soil was carrying into the environment to kind of help to kind of not only keep that bloom going, but to feed the biology that we want to keep promoting. So yeah, that's always, I think that's a great add to it, and you just take into account their recommendations, and you can scale it back. I don't think you need to go too hard on like sugar. You can overdo that type of thing to skew the population, but a little bit of trace minerals, a little bit of sugar, a little bit of protein to kind of help carry the biology for a prolonged period of time is a great add to the solution.
That's great. And Tom, thanks for that question. If there's not other questions, I did want to kind of ask one larger question to you, Misha, which is, you're looking at kind of helping vineyard owners and managers, transition more to a regenerative and sustainable agricultural systems. I mean, what are some of the trends that you're seeing in that? Are folks more willing to adopt those processes and practices, and are there kind of challenges that are holding folks back?
Yeah, I think everybody, like, it always sounds great. Regenerative farming means, more cost-effective farming and better return on investments. So, and it means less impact on the environment and preserving your soil to not lose it. You don't want to be dumping chemicals onto your soil only for those chemicals to get washed into a river, and it's, like you're essentially wasting your money and then losing your topsoil. I mean, that's your flagship of your farm is your soil. So if you're tilling it away almost literally by tilling it in the middle of the summer and dust clouds are going up and carrying your topsoil away, it's only a matter of time before, that negative impact will be exacerbated to a point where you're going to have to make some dramatic changes to restore it. So I think people are already seeing the symptoms of that. And as they start to try and adopt these things, these techniques and practices, they're starting to realize that it's going to be more of a push than they would like. I think everybody wants just some cure in a bottle that'll fix all their problems, but the reality is, like everything from the size of the equipment that we're using, whether it's a 25,000 pound machine versus a 500 pound machine. I mean, we got to start really taking a step back and looking at like what are the benefits of this, of the equipment we're using, what are the benefits of the products we're using, and how can we farm better. So those are really the challenges that are, you
Hmm.
know, telling a farmer he's going to have to swap out $300,000 worth of equipment is, it's just not that feasible. So how do we start with small changes and work towards some of the more dramatic changes that need to be made to make farming more sustainable is really where the challenges and the trajectory is going.
Thanks, Misha, and really appreciate you taking the time here. Again, you've been very generous to work with us on this, so we really appreciate your being an innovator and kind of working with us. Again, Misha is independent, works and runs his own crop consultancy. So I'll give you a little shout out. So you've been great to
Yeah.
work with us, and also, frankly, we've learned a lot on how to be better, on how to build this product better for farmers. So appreciate you doing that. Again, also just mention here the Biomakers soil tests are that's a third party company that we've worked with for years. Those are also things that are easy to access for farmers, and of course, Misha can help with that. But really appreciate you taking the time here. Is there anything you'd like to leave folks with as a final thought?
Yeah, just as we keep moving forward and trying to find solutions to growing problems that are persisting in the vineyards, I think that it's essential for farmers to keep an open mind and be willing to make minor adjustments to their programs because, if something's not working, or even if something has worked, 1,000 times, it's only a matter of time before it stops really working. And I feel like a lot of farmers are chasing that unicorn crop they had 5, 6 years ago or whatever, and they're too cautious to really adopt new products when, the anomalies that drove that particular success, unless you were measuring every data point, which is borderline impossible. You really got to be willing to venture outside of your comfort zone to find new avenues for success and really find cheaper solutions for what you need to achieve your goal, and really get a solid return on your investment and keep your farm alive and moving forward with the changes that the environment's throwing at you.
Right. Change is the only constant. Well, Misha, again, thank you so much for taking this. I appreciate everyone who's joined. And for folks that might watch this in after the fact, appreciate the willingness to engage in this. So with that, we'll kind of leave folks. I know it's a busy time when it comes to harvesting and to crushing. So looking forward to chatting with folks afterwards. So again, thanks for the time. Again, thank you to Misha, and we'll let everyone get on with their day.